Fopius arisanus is an important biological control agent in Hawaii because it attacks Tephritid fruit flies, which are serious economic pests throughout the tropics and especially here in Hawaii. The beauty of Fopius arisanus is that it attacks the egg stage of the pest insect, thereby controlling the insect at an early stage. In the following clips, you'll be able to see close-ups of how we rear Fopius arisanus in a laboratory setting, and at the end of this program you'll see some discussions of how this particular parasitoid has been used in a successful release program in French Polynesia.
We will begin our protocol with adult parasites that are ready to oviposit into eggs of a host fruit fly. The first step is to prepare the fruit fly eggs for presentation to the parasite. Next, fruit fly larvae are allowed to hatch, develop, and pupate. Once the fruit flies reach the pupal stage, they are sorted by size to enrich for parasites. Finally, parasites emerge and are maintained until maturation. They then mate and are again ready to oviposit into a new batch of fruit fly eggs. This completes the cycle.
Prepare a substrate for the fruit fly eggs by making agar blocks approximately 10 centimeters per side. The agar is prepared at a concentration of 9 grams of agar powder per liter of water. Using a level surface, fill the dishes to the rim with liquid agar, allowing it to solidify for at least 45 minutes. Once the agar blocks are solid, apply a single layer of tissue paper covering the top of each agar block. The tissue should stick easily to the agar because of surface moisture. Apply the fruit fly eggs to the tissue-covered surface of the agar blocks. A 0.5 milliliter volume of the fruit fly eggs in suspension yields approximately 6,000 eggs per block. A 2.5 centimeter wide paintbrush dipped in water is used to spread the fruit fly eggs evenly over the tissue surface. The fruit fly eggs are now placed under the bottom screen of the parasite cages to allow oviposition by the parasites. Tapping the underside of the screen removes dead parasites which might obstruct the fruit fly eggs. Allow the parasites to oviposit overnight. Approximately 21 hours is sufficient for a high percentage of the fruit fly eggs to be parasitized. Longer exposure risks superparasitization. The main components needed for rearing the eggs after exposure to parasites are fruit fly larval diet containers, and pupation containers. Place 3 to 3.5 prepared agar blocks of parasitized eggs on 1 1/4 liters of fruit fly larval diet. Place the diet container with eggs on a riser in a pupation container over a 1.5 centimeter deep layer of fine-grade vermiculite or washed sand. Tape the edges of the pupation container with masking tape. Ensure that there is sufficient space between the top of the diet container and the pupation container lid for larvae to crawl out and pop into the vermiculite below. Move pupation containers to a room at 27 degrees centigrade and 80% relative humidity for 1 week. Cover the containers with a heavy cloth for the first 4 days. After 1 week, open the pupation containers and remove the larval diet. We use Bactricera dorsalis as the host fruit fly to rear Fopius arisanus. Other species in the genera Anastropha and Ceratidis are also potential hosts. If there are unpupated larvae in the vermiculite, allow them to pupate overnight. Sieve the pupae from the vermiculite. Start with a coarse sieve in a large container. Next, use a hand sieve to remove any remaining clumps of vermiculite. Puparia containing Fopius arisanus can be partially segregated from unparasitized fruit flies by size. Based on previous research, we collect only the puparia between 1.65 and 2.26 millimeters in diameter. Puparia which are smaller contain mostly undersized parasitoids. Larger puparia contain mostly flies. A variety of methods may be employed to size sort the pupae. We will describe a mechanical method using the custom size sorter. For the collected parasitized puparia, the percentage of females increases with size. Place pupae in the funnel at the top of the mechanical size sorter. A vibration feeder moves them to the top of a pair of slightly divergent rolling bars at a 30-degree angle. The puparia drop individually into an array of slots that empty through funnels into 10 separate containers. Put pupae in the size range from 1.65 millimeters to 2.26 millimeters in an emergence cage for 7 more days until most flies have emerged from the unparasitized pupae. Next, use a fan to separate empty fly puparia from those still containing live parasitoids. To maintain the colony of Fopius arisanus, use a parasite holding cage with a removable glass front, approximately 25 centimeters in length per side. The cage has a 1 square millimeter screening on the top and 2 of the sides. The back of the cage is covered in a rubber sheet with a 9 centimeter hole for access. The bottom of the cage has a cutout covered on the inside with a 1 to 2 square millimeter screen large enough to allow placement of 2 auger blocks with fruit fly eggs for parasite oviposition. The holding cages should also have a tinted portion along the bottom quarter of the glass front to minimize parasite crowding along the front of the cage. Place approximately 11 grams of the selected puparia into plastic containers approximately 9 centimeters in diameter. This should produce about 600 to 700 parasites per cage. Size selection should result in about 60% females. Attach screen lids to the containers. The lids should be covered with a 2 square millimeter screening, which will allow Fopius arisanus to leave the containers upon emergence, but will contain any remaining fruit flies. Maintain the parasites at 24 degrees centigrade and about 45% relative humidity with a 12-12 photo period and good ventilation. Streak spun honey along the top of the cages at least 3 times per week or whenever the available honey is dry. Apply the spun honey in narrow streaks using a fingertip. Note that if the streaks are too heavy, the honey will drip into the cage. If the streaks are too light, the honey will dry out quickly and require frequent application. Place auger blocks which are 10 centimeters square and 4 centimeters deep on the tops of the cages to provide moisture to the parasites. These blocks should be changed 2 times per week.
In 2001, we received a small seed grant to see if we could introduce biological controls into French Polynesia to help alleviate the situation, because oriental fruit fly at that time was just causing unbelievable infestation of all the tropical fruit. The laboratory up here in Hawaii, we we made 10 shipments. So each one of these shipments usually amounted to about 100,000 Fopius arisanus. That actually it was quite a few more, but we would ship the oriental fruit fly pupae with the parasitoid inside, and then we'd emerge them. So we set up a small laboratory in Tahiti, and we would receive these, and then we would emerge the Fopius arisanus. So over a period, maybe of about a year, we released close to 1 million Fopius arisanus. Overall, this is the most successful, outside of Hawaii, when Fopius arisanus was initially released in Hawaii. It has been the best example of biological control of fruit flies throughout the Pacific. Over the last 10 years, there's been a tremendous spread of the Bactrocera dorsalis complex around the world. So Bactrocera caramboli has spread to South America, Suriname, and it's in northern Brazil now. Then there's been another species called Bactrocera invadens, which is spread throughout Africa and is causing all sorts of havoc. And then there has also been the spread of Bactrocera papayi. So this success now opens up opportunities to release these Fopius arisanus in South America, Africa, and in other parts of Asia.